Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. So, this is lecture 47. So, where I will briefly cover artemisinin, hyperforin and taxol, the three promising candidates for biotechnological exploitation. So, concepts covered will be artemisinin, hyperforin and taxol. These are the molecules of pharmaceutical interest. So, artemisinin is basically a triterpene. So, we have studied the terpenoid biosynthesis. So, there I have mentioned about this monoterpene, sesquiterpene, diterpene, triterpene. So, with artemisinin what happens that two sesquiterpene unit joins together and finally form this structure, and this is found in the plant of Asteraceae family, which is called wormwood, or the scientific name is Artemisia annua, and artemisinin is a very important compound because it is now used as an antimalarial drug. So, so the purpose of this whole class is just to give you an overview. So, here I am not going to do a detailed board work or explanation, so that you can get an idea, that is the only thing. So, do not get afraid about the structures and all these things. So, this is just to give you the concept. So, as you see that farnesyl diphosphate, so the FPP, and which is the first enzyme is basically the terpene synthase. So, it is also called this amorphadiene synthase as well, and that leads to the formation of this amorpha 411 diene, and then by several cytochrome P450 enzymes, the structure undergoes different transformation and eventually double bond reductase, and it forms the dihydroartemisinic acid. Dihydroartemisinic acid got converted into artemisinin. This is basically a non-enzymatic conversion. So, it is showing here dihydroartemisinic acid, then this is basically auto oxidation, which makes to artemisinin. So, what we see here the ADS is basically the what I have shown the terpene synthase. So, amorphadiene synthase. So, that is why it is called ADS. ADS is an important enzyme. So, this enzyme has been characterized at the molecular level, and several attempts were made to overexpress this, and as a result of this what happens to the final product. So, amorpha 411 diene by subsequently by cytochrome P450 and then reductase, it forms artemisinic alcohol, that will be converted into an aldehyde by a dehydrogenase, and then in one way it goes to artemisinic acid, another dehydrogenase, or there will be another reductase which makes dihydroartemisinic aldehyde, and then another dehydrogenase makes this dihydroartemisinic acid, and then finally it makes artemisinin. So, from the artemisinic acid it makes artenunin, and but from this one dihydroartemisinic acid it makes artemisinin. So, this is in general brief the biosynthetic pathway, and this is basically the same thing, only it is more clearly showing the product formation. So, I can skip this one. And other important point, as you know that sesquiterpenes are basically localized in the cytosol, as this is a triterpenes originated from sesquiterpenes coleton. So, most of the pathway is basically operating in the cytosol, and one important point here I have written here is the trichome. So, basically the leaf contains this fine hair like structure, what I have mentioned in one of the previous classes when I talked about the biosynthesis of menthol, and there I said that menthol and other products accumulate in the trichome. So, here also this products finally it accumulated in the trichome of this Asteraceae plant, which is Artemisia annua. So, as you see the most all the steps are all basically occurring inside the cytosol. So, that is the concept you should know, and you should know what role trichome plays. Okay. Now, the pathway and construction for engineering of artemisinin production in tobacco. So, tobacco was chosen as a model to engineer the pathway, and the pathway what is for the formation of artemisinin is shown here on the top, as you see here, this is the one written in red. So, these are the genes which were basically transferred into the tobacco, and that finally leads to the formation of artemisinin. So, the gene construct is shown here, and the other one is the pathway which is shown here. So, the work published in Nature Biotechnology in the year of 2011. That means, the take home message is that yes, it is possible to produce artemisinin in tobacco, provided you transfer the whole cassettes into it. And this is basically the experimental result, that indeed artemisinin is produced or not. So, they have checked the formation of artemisinin by using this UHPLC based methods. So, where you see that where it is this ADS transgene, here the detection of artemisinin is there. So, another transgene detection of artemisinin, whereas in the control or the transform plant lacking the gene, see only the empty vector. So, their artemisinin content is very less, and here is the same thing, they have checked it with the standards. So, okay. Next important point is that attempts were made to express this pathway in the yeast system. So, in the yeast system, these genes, that is the reductase and cytochrome P450 genes, several 2 or 3. So, these genes were transferred into yeast, and as a result of that, what we see that the formation of artemisinic acid. So, artemisinic acid was detected. So, artemisinin was not, but artemisinic acid was detected. So, that served as the precursor, and then subsequently using a semi synthetic approach, artemisinin can be made. So, here is basically the GCMS chromatogram, which is showing the product formation from the transgenic yeast, as well as only the control, and as well as the artemisinin found in the Artemisia annua plant. So, this is also published in Nature. So, that means take home message is this, yes, it is possible to produce the ultimate precursor for artemisinin in yeast. So, from here I move to hyperforin. So, hyperforin again it is a it is basically we can group it under terpenes, because there are a lot of prenyl chains attached to it, and it involves this DMA PP as well as GPP, you will see. Okay. So, what is basically hyperforin? Hyperforin is bicyclic polyprenylated acylfluoroglycinol. So, this I can write it for you. So, it is basically bicyclic polyprenylated acyl fluor PHLO fluoroglycinol derivative. So, this is what is called hyperforin. So, hyperforin is a very very important molecule, because it is a proven antidepressant and it is safe to use. Therefore, in the European market, this is in the herbal shops, it is sold at as St. John's wort. So, St. John's wort is available, it is available over the counter. So, anyone can buy it and use it, and this is the plant. In the pistil the hyperforin accumulates, but apart from that, in the petals there are tiny dots which are reddish in color. So, their hypericin accumulates. So, I will show you some of the nice pictures. Now, pharmacological activity is basically one most important point is antidepressant, that is why the molecule is so important, but apart from that it has anti-inflammatory, antibacterial, neuroprotective, anti tumoral, antimalarial and so many. So, we are not going to discuss these things, only to tell you that how important is this molecule. Now, because of its importance, so scientists they wanted to produce this product in cell and tissue culture approach, so that all round the year the plants can be grown. Otherwise, and moreover this is specific to temperate region only. So, in the tropical area these plants cannot grow. So, if you have a culture system, then it is possible to use biotechnological intervention to produce this. Now, these are the leaves with these dots, and this is so, and here a mature leaf, it is basically red in color, and this red color is because of the hypericin accumulation. So, and the translucent dots are also there, where the hyperforin accumulates. So, structurally these are different, as you see there are a lot of phenolic rings joins and form hypericin, but that also formed by polyketide synthase activity. Hypericin is also by polyketide synthase activity. So, that way there is a relationship between these things. So, dark nodules accumulate hypericins, whereas translucent gland accumulate hyperforins, what I said. So, this is for the hyperforin and this is for the hypericin. So, root cultures of Hypericum perforatum was established, and these root cultures were shown to accumulate hyperforin. So, this is the plant and this is the root culture, very interesting. And then cell suspension cultures of a related species was also used to study the biosynthetic pathway, and their cell suspension cultures were treated with methyl jasmonate, I have mentioned in the elicitation class. And then what happened, that upon elicitation it accumulates hyperxanthone, not specifically hyperforin, but that itself was appeared to be of pharmaceutical interest, and that is why I mentioned here that cell cultures can also upon elicitation produce different interesting compounds. Now, the proposed biosynthetic pathway is basically like this. So, here what happens, that 3 molecules of malonyl CoA, this is malonyl CoA joins with isobutyryl CoA. So, this is basically isobutyryl CoA. BUTYRYL isobutyryl CoA, this is malonyl CoA, and it forms this fluorine is a big name, this one I write it here, PHLORIS fluor isobutyrophenone. So, which is the product, and this is by the enzyme, again it is a polyketide synthase. So, PKS, or subsequently this is PKS means polyketide synthase. When we talk about phenolic compounds, we will mention about this in little bit more details, and here specific polyketide synthase has been identified, and this was termed as BUS. So, BUS stands for isobutyrophenone synthase. So, this was reported in the famous journal Phytochemistry by Professor Ludger Beerus and his team, and the paper published probably in 2005. Okay, I have okay, sorry, I have not put the reference here, but reference can be provided later on. And next step is basically the prenylation. So, this is issue the prenyl chain 1, 2, then 3. So, and you see here DMAPP is required, first one, second one again a DMAPP, third one is again a GPP, and the fourth one again a DMAPP. So, basically these are all prenyl transferases. So, these steps are all catalyzed by PTs or PT, you can say prenyl transferase. So, which transfer the prenyl side chain into it. So, the first prenylation step is catalyzed by a soluble dimethyl allyl transferase. So, here basically this appears to be a soluble enzyme. Normally, prenyl transferase are membrane bound enzyme, but the first one what they found to be a soluble enzyme. Okay. And the major constituents accumulating in hypericum perforatum, which includes hyperforin, hyperesin along with several xanthones, which are all the polyketide derivatives. So, this is in brief the biosynthetic pathway, which has been characterized only at the biochemical level, okay, not at the molecular level yet. It is very complex. And then hypericum perforatum in vitro root cultures were established. This is in vitro grown plant, and this is the root cultures, and then root cultures are grown, and a fully grown root cultures here, and these are basically the freeze dried root cultures, which are used for subsequent extraction and analysis. Okay. So, hyperesin production was also checked from this root cultures established. So, this is the normal plant, and in vitro grown plant, from their callousing and then from root formation, then roots started growing, and this is the fully grown roots, and then this is basically the dried roots, freeze dried roots like stack coins. Okay. And then what they have done, they have compared the hyperesin production in shoot cultures and root cultures. So, the shoot cultures is basically shown in the red is from the shoot cultures, and the blue is from the root cultures. What you see here is the secohyperforin, which is number 2 I have marked, and then hyperforin number 6. So, here you see that the production of hyperforin is higher in root, okay, whereas secohyperforin is more in the shoot. Okay. And number 8 is basically the adhyperforin, which is almost undetectable in the root. Apart from that, there are other compounds like number 5, which is more in the root, okay, also number 1, which is also more in the root. So, this indicates that root cultures are indeed very promising for the production of these compounds and for subsequent study. With this, now I move into the taxol. So, taxol is a very important anti-cancer drug, particularly for treatment of ovarian and breast cancer. So, this is the figure of the bark of Taxus brevifolia, the original called yew, Pacific yew tree. So, these barks are harvested, and what is important that 5 to 600-year-old trees are required to treat one patient. So, the amount of this taxol is so low that you require so many trees. Subsequently, they found that actually Taxus brevifolia is restricted to certain areas. So, but another species which is more widely available is Taxus baccata. So, and scientists also found Taxus baccata is also accumulating taxol. Not only that, much later they found that the Taxus needles are also accumulating taxol, although in low amount. And of course, much later they found certain endophyte taxomyces, which are basically living in the soil where taxus are growing. That species are also capable of producing taxol, although the content is less, and then lot of efforts are being made to make taxol from the microbial sources. Now, one important point is this: the taxol, as you see here, the registered trademark is there. So, the drug is called taxol, but the original chemical compound is basically paclitaxel. So, the paclitaxel is the chemical compound, taxol is the drug. Similarly, the docetaxel is the generic name and the drug is called taxater. So, that I will come. Yeah, it is written here. You see the registered trademark is there. So, this is basically produced by Rowley-Poulenc. So, the product is taxotere, but the original molecule is docetaxel. Similarly, taxol is produced by Bristol-Myers and the product is paclitaxel. So, this you must know. So, when you say taxol, it is not correct, either we should tell paclitaxel or taxotere or docetaxel. Yeah, but we are more familiar with taxol rather than paclitaxel, that is our problem. Okay. And so, this is basically the structure of taxol. You see how complex the molecule is. So, it is basically again this terpene derivatives and this is the taxotere. Okay. And what is showing here in the left side is a 3-dimensional structure of taxol, and this is basically the taxol, the taxane ring is here and the ester side chain, which are basically important for imparting the cytotoxicity and unique effect of microtubules. Okay, this will tubules is an extent tubules. So, okay, I will check it. And as harvesting trees are not very feasible, so the scientists they look for alternative sources for taxol taxater production. So, one is the cell cultures, and cell cultures particularly using this coronatine as elicitor, that showed promising effect in producing paclitaxel. So, I will not go into details of it, and little bit of biosynthesis. So, why because it is basically starting with the GGPP, generalized pyrophosphate, then there is a taxane synthase and then it produces this taxa 4,11 diene. Similarly, what we found this amorpha something diene in case of artemisinic acid, then several intermediates and then it finally forms baccatin. So, baccatin is an important precursor and from baccatin paclitaxel is formed by the joining of this from phenylalanine, the phenylalanine CoA will be formed and that it joins and ultimately it makes the paclitaxel. So, and diacetyl baccatin, from baccatin diacetyl baccatin is formed and that is used as the source for semisynthetic different derivatives of taxol production. So, this is taxol, this is taxotere, this is diacetyl baccatin. So, baccatin is important and the biosynthesis is very complex, although biosynthesis has been has been resolved mostly, chemical synthesis was also, but producing paclitaxel chemically is not feasible. Okay. And that is why scientists are looking for biotechnological approach, first with the empirical approach by cell and root cultures to produce that, and later to still yet years to come, and when there will be a time when maybe the pathway can be expressed in yeast, and then what we have seen in case of other alkaloids, that maybe another 10 years or so that de novo production of taxol in the yeast may become a reality, but still now it is not, we have to wait. And you see there are multiple enzymes like synthase, then hydroxylase, then acetyl transferase, then again hydroxylase, so many hydroxylases there, and these hydroxylases are mostly membrane bound. Okay, then oxidase and then again acetyl transferase. So, this forms the baccatin, then from and then it forms other products and eventually the taxol. So, this indicates how complex is the biosynthetic pathway, but it is very interesting and lot of work has been going on. So, years to come we will see some more outstanding achievements. So, with this I end this class, and with this I have given you a brief overview of artemisinin, which is a very important antimalarial drug, hyperforin, which is basically a very strong antidepressant and safe antidepressant, and third one is the taxol or paclitaxel, so which is basically a proven anti-cancer drug against ovarian and breast cancers. So, with this more or less I finish the terpenoid group, and the remaining classes I will concentrate on the phenolic compounds. Thank you very much.